Okay, so good morning, everybody. Thank you so much to join this lecture. First, I want to thank Amar and Federico to support the idea and help in this presentation. The idea was to prepare a lecture on nanobiotechnology and how it could provide some solutions for this pandemic COVID-19 that we are experiencing. There are two objectives. The first objective for the student is to actually link or connect from what they are learning in the classroom in this class of nanobiotechnology with Amar and Federico and myself to the reality, the current reality that we are facing. And the second is to maybe generate some ideas, generate some curiosity about nanobiotechnology and possible impact. Okay. The main this lecture was prepared in a way to cover three main aspects. The first aspect is prevention, diagnostic, and therapy. And of course, in all these three aspects, there is a contribution of nanobiotechnology, and this is what I will review today during this class. So the way this class was prepared is to look at all the publication or press that have been reported talking about nanobiotechnology and coronavirus. Of course, there's a lot of things here that are probably missing. Maybe some ideas that could be used for prevention, diagnostic, or therapy of coronavirus. But here is really focused on what has been tested with this virus or with a virus that is very similar to this virus. Okay, so let me start by the first slide, which is really going to put all the details that I'm going to talk about in this lecture into perspective, okay? There is an emergency, and this emergency has resulted in the need to, in most cases, recycle ideas, recycle technologies that in the past have been shown to work, but they have not been shown to be needed. There was no need. It worked, it's great, but we did not face such a serious pandemic that resulted in the need of many of these technologies. So if you look in this slide, you can compare the SARS, right, that came up in 2002. I'm sure some of the professors probably remember this time. I do remember well when I was in Singapore, because Singapore was really affected by SARS. And then, of course, this SARS-CoV-2, right? Hold on. So you can see the difference. You have, for the SARS-CoV, you had about 8,000 cases. For the SARS-CoV-2, you have more than 50,000 cases. Of course, this information changes every day. It's much higher than that now. But you can also see that the number of deaths is much higher. And in terms of countries, pretty much is the same number of countries that have been affected. The mechanisms of these viruses are pretty similar. They can infect humans and they also infect animals. They have some common biological factors, and this has been well demonstrated. Okay. Now, if you look on the right side, you have the number of cases and number of deaths for SARS, for MERS, and for this recent coronavirus. And you can see the scale, okay? The scale for this coronavirus is extremely aggressive. It's a coronavirus that can, it's the virus that can infect very efficiently. And one of the main reasons is because for 2 weeks, there is no symptoms in humans. So people can actually travel, continue their daily activities, and contaminate people for 2 weeks without knowing that they have the virus. Now, if you go a little bit below, you can see that MERS took about 900 days or 2.5 years to infect 1,000 people. This coronavirus took about 48 days. So extremely rapid infection. And due to this emergency, there is a lot of technologies, a lot of products that were not used for SARS or for MERS, but now could be used, okay? So the first part of this lecture is prevention, and I will start by these nano antimicrobial or nano antiviral disinfectants. Okay? The idea is to spray, to coat, basically to change the surface where there is a possible binding of the virus and kill the virus, okay? So on the right side, you can see a table of a list of nanomaterials, mostly inorganic nanomaterials that have been shown to be antimicrobial and antiviral, okay? One of the most popular is, of course, copper oxide, CuO. This is a material that is very cheap and has been shown to be a pretty good antimicrobial, antiviral disinfectant. Now, how it works, you can see on the bottom of this slide. Typically, these inorganic nanomaterials can kill bacteria and viruses in 2 ways. They produce reactive oxygen species that can create oxidation stress in these organisms, and then they can either break the membrane or they can also bind to the DNA and induce death, okay? Okay, so you can see this man spraying the floor. You probably have seen this everywhere. There is a company in Italy that has proposed to spray, to coat buildings and roads with copper oxide surfaces, nanoparticles. Now you may ask yourself, what is the advantage of copper oxide compared to bleach, for example? Because we know bleach can also kill this virus, right? And we use it at home. Well, one advantage is that copper oxide can be active for a long period of time. So you could need maybe one spraying, one coating, and then you could keep it that way for a long period of time. If you use bleach, you probably have to do it every day, maybe multiple times per day. And this is a disadvantage of using bleach, although it works. So now all this work on using copper oxide nanoparticles have been based on these studies that show that this virus, this SARS-CoV-2 virus, and also the first one, are not very stable in copper surfaces. You can see on the top. So here you can see this, the number of hours where the virus remain alive. And you can see that actually this virus is very resistant in aerosol, okay, over time. Now, if you do the same thing and you put the virus on the copper surface, you can see that this virus start to die very quickly, okay? On cardboards is also not so stable, but it still takes some time. Stainless steel is quite stable and plastic is quite stable, okay? And actually, if you look at the bottom here, you can see that the slope of this curve is much higher than the slope of all these curves here, which suggests that these copper surfaces can actually efficiently kill this virus. And this is the beginning, this is the result that really demonstrate that copper surfaces, copper nanoparticles, could be very useful to prevent binding or infection of this virus. Now, this is a product that was reported, and you can see here they actually develop this mask. We have seen this mask, we use them probably every day. But in this case, what they did is that they used the fabric of the mask and then they grow a polymer, okay, on the surface of this mask. And then with this polymer, they can bind these copper oxide nanoparticles, okay? And if you look at the electron microscopy, you can see that here you can see these copper nanoparticles in the fabric, okay? So the idea of this work was to use these nanoparticles into a mask so that not only the virus can be captured by the mask, but the virus can be killed by these nanoparticles. And again, based on this copper oxide activity, okay? The next one is quite similar. This is a product that actually has been available for some time, RESP mask. And you can see on the right side here, they compare the characteristic of RESP mask compared to the N95 mask that probably most people are using right now. And what is interesting is that this N95 mask has a pore size of 250 nanometers. Yeah. Whereas this RESP mask is made of nanofibers, and now the pore size is much smaller. It's about 25 nanometers. Okay. And this is basically the structure here of this RESP mask. You have a top layer, and then you have the nanofiber layer, etc. Okay. Why this pore size is important? Actually, it is very important. And the main reason is that this electron microscopy of coronavirus has been published. And if you look at carefully the scale, you can appreciate that this virus is much smaller than 250 nanometers, right? This virus is about, this is 50 nanometer scale. So this virus is about 150, right? Maybe less than that. So this could really support the need of a mask that have a much smaller pore size to prevent the spreading of this virus. Now here's an electron microscopy of actually the fibers of this mask. And then you can see here all these fibers that can form the fabric of this mask. And this can prevent the virus to pass through because the pore size is much smaller than the size of the virus. Okay. Now, in their latest development of the mask, they have used copper nanoparticles as well. Sorry. They have used copper nanoparticle as well, and it is the same principle. The principle is to capture the virus and also to kill the virus, okay? Using these copper nanoparticles. So again, this is a product that probably in the past we will never really appreciate because there was no need. If I remember well, during the SARS in 2002, 2003, I don't even remember that people were wearing masks. At least in Singapore, we were only, we were not allowed to travel out of Singapore. But I don't remember people wearing masks. But now there is a need because there is a serious health and financial situation that need to improve very quickly. Okay, so this is another mask, and here he explained a little bit. Sorry.
I have a question. What happens if they are in contact with the water?
Yeah, Maria, I think what would be best is that I will finish the PowerPoints. Please write down your question on the chat, and then I will answer all the questions in order in the chat so that I don't miss any questions. Okay, that will work well, and then maybe we can talk if there is something that is not clear. Is okay for you, Maria?
Yes, yes, okay, teacher.
Excellent. So there is also this nanoenzyme concept to deactivate coronavirus that was reported. And this is quite interesting because here they really shown the mechanism of these copper nanoparticles. What happened is that these copper nanoparticles can bind to the virus, okay? And when they bind to the virus, they can react, and when they react, they actually create this lipid peroxidation. What does that mean is that there is a reaction on the lipid membrane of the virus. And when this happens, this lipid membrane breaks because now the lipids are not stable anymore. Okay? And if you see on the right side, here you see this virus. And then if you increase the concentration of these copper nanoenzyme particles, what do you see? You see this breaking of the virus membrane. And because of that, right, now this virus cannot infect cells anymore, okay? So not only they have shown that this nanoenzyme can actually break this surface membrane of this virus, they have shown that when this happens, there is a production of radical species. And these radical species can react with the proteins of the virus. And they actually start to degrade the protein of the virus, just like bleaching a protein. And if you look on the right side here, here is the increasing concentration of this copper oxide nanoparticle. And then you can see this band here is the presence of proteins. This is the protein of the virus. But you can see that these three proteins disappear. That means that they have been degraded when they interact with these copper nanoenzyme particles. So they have developed this mask and tested this mask. On one side is the control without these nanoenzyme particles. And on the right side, the nanoenzyme, okay? But again, it's kind of the same principle as before, using nanomaterials to either capture this virus, degrade it, or do both. This is one of the last reported masks. And in this case, they are actually using graphene nanomaterials in the mask. And this company is selling this mask, you can see here. And the materials in this mask is made of graphene. And now they have compared the surgical mask versus the this Guardian mask, this graphene mask. And you can see that they have similar properties, but here, this graphene composed base mask can actually have antibacterial activity. They can trap bacteria and viruses, etc., etc. So they have some advantages compared to the regular mask, okay? And why is that? The main reason is it has been shown that, for example, bacteria that is mixed with graphene oxide, okay, without graphene oxide, bacteria look like this. If you increase the concentration of graphene oxide, this is what happened to the same bacteria. And what you see here, you see a change of shape, right? And this change of shape means that this bacteria is actually dying. And here you can see the bacteria is completely broken down. And how does it work? It works because this graphene is negatively charged, and then this graphene interact with positively charged of bacteria or virus. And when this happens, because graphene is a very thin layer of material, it actually can puncture, can break here the membrane of the virus. And when this happens, the virus can die, okay? So this is a summary of the prevention method. And you can see overall, most of the work in prevention has been done in surfaces, in material chemistry surfaces, and it has been adapted to using mask, protection mask, okay? Now let's look at the diagnostic aspect. And here is the rapid point of care detection test of these two antibodies, IgM and IgG, that are present in serum. You probably have seen this device everywhere, okay? This is a quite cheap device, and this is how it works, right? It's a very small device, and here you have an inlet, okay? In this inlet, first you put a solution, and in the first solution is a buffer. It's basically water, and there is a control antibody inside, okay? A biomolecule that is used in the buffer as a control. And then after that, you take the blood sample, you put it inside here, and then you wait. And when you wait, you can have 4 different results. This result is negative. Everything is okay. If you have these 2 bands, that means the patient in his blood has the production of IgM antibody. I will explain a little bit why these 2 antibodies are important for this virus, okay? And then if you have this middle band, that means it's IgG positive, yeah? Now, if you have the 2 bands, okay, that means it's IgM and IgG positive, right? Now, you can observe here that you always have this band right here. This is the control band. It tells you if the test worked or if you can trust the test or not. So if somebody do a test and this band does not appear, but this band appear, you cannot report the result. That means something happened during the test and the device did not work well, okay? Okay, so I explain a little bit this. Now, why IgG, why IgM, and why IgG, okay? During a viral infection, you have, your body will produce 2 types of antibodies. The first one is the IgM, and the second one is IgG. So these are basically biomarkers in your blood that suggest that you have been infected by a virus, okay? This is not specific to COVID-19. This just tells you that you are infected with virus, okay? That's why it's called rapid point of care. It does not give all the information, but it tells you if you are at risk or not. Now, typically, IgM is produced early during the infection, and IgG is produced a little bit later in the infection. So if you have one band or two bands, it can tell you how, it can give you a timeline of infection, okay? Okay, so now let's go, I still didn't explain to you how it works, how nanotechnology are involved in this device. So this is the way it looks like. You have a sample pad, you have a conjugated pad, and I will explain exactly what you have in all these different pads. And then you have three lines. You have the M line, G line, and then the control line, okay? And this is made of paper, right? It's a nitrocellulose membrane, so it's paper-based. Now let's look a little bit what it is in each of these part of this membrane. So the first is the sample pad, right? This is where you actually put your sample with your control. And here you have a capillary diffusion mechanism. So your fluid is going to diffuse from here to the other side of this paper-based device. Now, in this conjugation pad, okay, you have gold nanoparticles. Gold nanoparticles that are modified to bind to the antibody of the COVID-19, okay? So if the blood sample pass here, and this sample has IgG or IgM or IgG, they will bind to gold nanoparticles. If it does not have this, the sample will stay in the solution. So it binds, now it continues to go here. And in this line here, okay, you have a different antibody. You have an antibody that now bind to IgG or IgM. In this case is M. So here you have a line of all these antibodies, you can see here is in the picture, that bind to this IgM. And this is important because when it binds, the gold nanoparticles stay in this line and they accumulate in this little line. That's why now you can see the change of color because of the accumulation of the gold nanoparticles. Here, you don't see it because these gold nanoparticles with the IgM do not bind to anything. Yeah, so the color is very light, you cannot see it. But as soon as it's in contact of the other antibodies, then they accumulate, they complex, and you can see the color. Now, if there is no IgM, this sample pass through and then now is in contact of the other antibody. And if there is IgG, now these nanoparticles accumulate in this little line and create this change of color again. And then you have the fluid that continue to pass. And of course, the control sample, the control antibody that did not bind to any of these IgM and IgG antibody will bind now to the control. And now you have a change of control or change of color, okay? So it's 3 steps. The gold nanoparticle will pass this, will diffuse, will flow and bind to the antibodies, accumulate, change the color for IgG, then continue, change the color for IgM, and then continue and change the color for the control, okay? And this can take about, I think if I remember well, maybe 20 minutes or 15 minutes, so it's quite fast. Now, as I say, this does not tell you that it's specific, okay? But I think there are some tests now that have a more specific antibodies. Okay, so I'm sure you have seen this test everywhere on the TV, and actually I think they are using this now in Ecuador as well. The other one is interesting. This is actually quite new. This group of scientists from Europe, Spain, France, have developed a reagent-free optical nanotechnology system as a biosensor. Now, this is specific to coronavirus 19, but here the key is that is reagent-free. So why this is very important? And why this is very important now? Why before we actually did not care about this? Well, before we could buy the reagents and they could be delivered maybe in 1 week or maybe in 3 days. But now these reagents to do most of the tests are not available anymore. Not only they are not available, even if they are, they may not be delivered quickly. So there is a need to develop methods that do not use reagents, okay? And this is the main impact of this method here. And they have reported that they need maybe 2-3 months to have a prototype ready. And they describe this prototype as a small little box, and that could be used to detect coronavirus. Now, how it works? Basically, you have this microfluidic channel. What is a microfluidic channel? It's basically a small device that allows flow of fluids, okay? And this channel is modified with molecules here that bind to this COVID-19. And when the COVID-19 pass in this channel, it will be slowed down. It will bound to these proteins because this protein will capture this virus. And when this happens, you can put light and it can affect the phase of the light. And because of the change of the phase of the light, you can actually detect if there is a presence or not of the COVID-19 in the blood without using reagent. And here's a little bit how it works. You have a reference where the light passes through here. And this is important for the phase here. And then you have your sample path where the light passes as well, but now it will change the phase of the light. So you can see here light passing to the reference, light passing to the sensing. You have an objective, and then depending on the change of phase and the time, you can actually collect a different signal. And if it's flat, that means there is no virus. If it starts to have some waves, that means you have virus inside the blood, okay? So, next one is also a same principle, actually. Another reagent-free method. But in this case, is using magnetic nanoparticles for high throughput RTPCR analysis of this virus, okay? As you know, RTPCR, you have probably heard about this word many times in the past month. RTPCR is an instrument that can specifically identify the presence of the virus based on the genetic information. In this case, the RNA information of the virus. Okay, I will explain a little bit how it works. But the most important is to understand why, in this case, magnetic nanoparticles could be very useful. So this company in, I think is in Denmark, Sweden, I don't remember, have reported that now they can do 150,000 tests per week. Yeah. So in the past, we actually don't care how many tests per week you can do. As long as you can do enough tests to check the flu or some other viruses, is not a big problem. And therefore, this technique has not been really used too much. But now, doing a high number of tests in a very fast manner is actually very important. Why? Because every day there are a significant number of persons infected and death. So the more tests, the faster the test, the better the decision can be made to control or prevent this disease. So now it becomes extremely important. And that's why now many people are interested in this kind of technologies. Now, this group from China has published a paper using magnetic nanoparticles, and they basically explain the process, okay? Now, using magnetic nanoparticles to extract RNA has been reported more than 10 years ago. There is more than 20 papers reporting this. But the key is that for the past 20 years, not 20 years, 15 years, for the past 15 years, we didn't really have a use for it. But now we do. And this is the nanoparticles, right? This is the magnetic nanoparticles. And then they make a coating, okay? And you can see the charge change, okay? And then they do another coating. And now you can see that they make these nanoparticles that are very negatively charged, okay? So now they have magnetic nanoparticles that have a very high negative charge. And I will explain to you why this is important. Okay. Now, on the top here, this is the typical test done to measure, to verify the presence of the virus in the blood, okay? This has been used in pretty much all the diagnostic laboratories in the world to detect coronavirus, including here in Ecuador, at INSPI and in other laboratories. Basically, you take the virus solution and then you use a solution that breaks the membrane of the virus. When you break the membrane of the virus, you have the genetic material that can go inside the solution, okay? And here you can purify this RNA, okay? But typically the concentration of this RNA is very small. Typically you use very little amount, 10 microliters, 20 microliters. So what you do is you do a little bit of biochemistry, you expand this RNA and you create more copies of this RNA. And then now you can actually detect this RNA using this RTPCR. The RTPCR is very specific. So when you have a signal, you can see here, so let me, you can see here the RTPCR result. If it's negative, you have this kind of signal. If it's positive, you have a curve that starts very quickly and go and flat, okay? Now, when you have this, this is very specific to coronavirus. If it's another virus, it's not gonna show up, okay? Now, to do this reaction, you need a lot of reagents, different reagents, okay? To purify, to amplify, and all these reagents currently are actually not available. So why using magnetic nanoparticles is very easy. You could put the same lysis buffer and then you could collect the RNA into this tube. Now, because your nanoparticles are negatively charged, these nanoparticles bind to the RNA, okay? And now you can use a magnet. Here you can see you can use a magnet. So you can actually collect the nanoparticles at the bottom of the tube with the RNA. And then you can do the wash, and then you can either remove the RNA from the nanoparticles and do your analysis, or you can do the analysis directly on the nanoparticles. So you can see that this process here is much faster, much easier, because it does not involve all these reagents. You only need the buffer to break the virus, and you only need the nanoparticles to collect the RNA, okay? You don't need anything else. So, now I will go to the last part of this, this lecture. I will talk about therapy, okay? So, of course, you have heard a lot about the vaccine, the different clinical trials of vaccine. And before I get into the different therapeutic approaches, I think it's important to remember the different types of vaccine, okay? There are three main regions in the vaccine that are very important. The first one is the genetic material. The second one is the membrane material. Oops, sorry. And then the third one is whatever is on the surface of this membrane, okay? All right. Now, you can produce different type of viruses depending on what you are going to present or inject inside the body. It can be a peptide that is similar to the protein of this virus, okay? It can be a virus-like particle, so it could be a polymeric type of particle that has a genetic inside or only has the protein outside. So anyway, depending on what you want to focus on, is the genetic material or the surface of the virus, you can create or you can use different type of virus. Yeah? Now, if you look at the landscape of viruses that have been explored or tested, okay? Here is the list of the different type of viruses, okay? And here you can see this is the exploratory confirmed. What does that mean? That means that there are studies that reported using these kind of viruses for coronavirus, but they are still early stage, but they have been confirmed. And then you have a group that actually is not confirmed, is the data is probably not available yet, okay? And then you have a group that is in preclinical. What preclinical means is that they are now being tested in animals, okay? And then of course you have the last group that is in phase 1. Phase 1 means that is now tested in human. So first thing you can see is that from the groups that are exploratory to the group that are preclinical, you have less than half, probably the third, okay? So not many have been tested in animals. And now if you go from the animals to the human, you can see that again is less than a third or the quarter that are now tested in human. And you can see that actually the most common one is the inactivated virus type, okay? So this gives you an idea of all the efforts to develop vaccines and actually what could become a vaccine that will be used. Now, if you look at who is developing these vaccines, in everywhere in the world is mostly private companies and industry. All these viruses have been mostly developed by the private sector. But you do see a little bit of involvement of academia in some regions, okay? Now you probably also heard a lot about the drugs that could help to actually treat this virus. I'll give you a brief table, kind of summarize a little bit, but here is basically the list of the drugs that are now in clinical trials. Now, none of these drugs are new drugs. All these drugs have been used in some ways to treat other disease, okay? In fact, most of these drugs are antiviral drugs. They have been used for HIV, probably for some of them for malaria, etc. So the idea is that the mechanisms for viruses to infect the body is quite similar. So a drug that works for a drug that worked for one virus could also work for different type of virus, okay? But you can also see that they have been testing combination of drugs. And this is more and more the result of the clinical trials demonstrate that one drug probably is not enough for the treatment of this virus, okay? Now, of course, you've heard a lot about hydroxychloroquine, and I will talk a little bit about it. So hydroxychloroquine is a key molecule because actually it has been used in nanomedicine for a long time. And this drug has been used, why? Because this drug block the internalization of the virus inside cells. So people in nanomedicine have used chloroquine to study the mechanism of entry of nanomaterials inside the cells. So here in this graph, you can see that this virus bind to the surface of the cell, and then it can enter inside the cell, okay, through this complex of proteins here, these green proteins. Now, what happened with chloroquine is that this chloroquine blocks this complex formation. So the virus cannot enter inside the cell. So chloroquine does not kill the virus. Chloroquine prevents the virus to enter the cells and to infect inside the cells. Now, why? You heard about chloroquine and you also heard about hydroxychloroquine. The main difference is this functional group here. And actually, if you look at these two drugs, they can stay, both of them can stay a long time inside the body, many days actually. And also, they can have a very high concentration inside the blood. However, hydroxychloroquine seems to be more efficient at lower dose than chloroquine. So hydroxychloroquine has been one of the best candidates to prevent infection of coronavirus. So here's another strategy, right? This drug chloroquine, as I told you, prevent the virus to enter the cell. In this case, they have demonstrated that they can use a nanoparticle, okay, made of a protein, okay, and this is called a phage capsid, okay. This is how this phage capsid look like, and basically is a complex of protein units that make a nanoparticle. Now they can modify this nanoparticle with a functional group. See this? And what is interesting is that this functional group actually bind to the protein of the virus on the surface. And here you see actually this virus, here you see the virus, and in blue is the protein, and in purple is actually the phage capsid binding to the surface of this virus. And what is the result of this is that now the virus cannot enter inside the cells because it's covered on a surface by other molecules. The protein that help the virus to enter the cells are blocked. So it's very similar mechanisms as chloroquine, but instead of blocking the cell, now they block the virus itself, okay? And of course, in this case, you need nanomaterials to do this. Now, there is another approach, which is quite similar, actually. Instead of using this protein nanoparticle, they use this carbon quantum dot. And this carbon quantum dot modify with this functional group here can bind to the surface of the virus and again can block the entry of the virus inside the cell, okay? So same concept, but different kind of nanoparticle. Now this one is very interesting. Actually, it's probably one of the most recent technology, and this was developed by Ufovax. What is interesting in this nanoparticle is that they basically have some genetic material, okay? This genetic material is inserted into cells in the lab, and these cells can produce these protein units, okay? And these protein units have these proteins here that is called antigen. Antigen is basically the proteins that are on the surface of viruses that are recognized by body immune system. So these proteins here, these antigens, are very important for our immune system to develop the antibody and block this virus. Now, this vaccine, this company here can design these units to have any kind of antigen. So they developed these units here for coronavirus. And what is also interesting is that these units can assemble themselves to make a nanoparticle, okay? So they create this little block and then they assemble this block into a nanostructure. Now, of course, they have to purify. Why they have to purify? Because they have some free blocks, right? Not all the blocks can self-assemble. So they have some free blocks and they want to remove this. So they can remove this and then produce this kind of nanoparticle vaccine, synthetic nanoparticle vaccine. And these can be used as a immune system to stimulate the immune system and block this coronavirus. This one has been also in the news a lot. This is probably one that is one of the most advanced from this company, Moderna. And in this case, Moderna has developed a lipid nanoparticle, a lipid nanovaccine against coronavirus. And what is the difference between a liposome and a lipid nanoparticle? I kind of give you here the structure, okay? Typically, liposomes have a compartment here, and then you have bilayers of lipids very well organized. In the case of lipid nanoparticles, you typically only have one layer of lipid. Oops, sorry. You have one layer of lipids, and then you have some small lipids inside that can create smaller compartments. But the surface of this nanoparticle is typically made of one layer of lipids, but in liposome is made of two layers of lipids, okay? Now, what is very interesting about this is that they have developed this vaccine in very short time. You can see the scale here. This is when the amino acid sequence was selected, and then they designed the RNA, and then within 17 days, they were able to produce this vaccine. They only needed more than 2 weeks to produce this new vaccine. And then within 41 days, they were already testing this in clinical trials. So in a little bit more than 1 month, they were able to produce this vaccine and to test it in human. This is extremely fast, okay? As you may have heard, right? We will get probably, the first vaccine will probably be available in 12 months, 16 months, it depends on the clinical trials. But going from designing the vaccine to the human in 40 days is extremely fast. This is one that was recently actually in the news. And in this case, they actually reported the use of microneedle to deliver vaccine for COVID-19. You may be not very familiar with microneedles, but basically they are small devices, okay? And these devices have little, very tiny needles compared to the typical syringe. And the reason for this is that these needles do not go deep into the skin and therefore do not create pain, okay? Now they are using a vaccine that is based on the spike protein of the virus. And the reason why, so here is actually the microneedle here, device. You can see very small. You can take it, you can press it on the skin, and then this needle will pass through the skin and deliver the vaccine. And the reason why they're interested in this is because the immune reaction in the skin is much stronger than in the muscle. So the hypothesis is that they will be able to create a good vaccine if they can deliver this inside the skin, okay? And they actually have animal studies that show that it can work. So with this, I'm just gonna thank you everybody for your time. And I will be happy, I don't know what time is it, but I will be happy to answer some questions. Okay, so I have the question from Maria Belen. Okay.
May I make a question? I can make a question?
Yeah, please, it will be good if you write it so I can follow the order, and then if there is need, we can also talk. Yeah.
Sorry?
So the first question is from Maria Belen. And hold on. Yeah, Maria Belen. Okay, what happened with the nanoparticle when masks are in contact with water? Okay, so it's a good point, okay, Maria Belen. Depending how the nanoparticle are integrated into the mask, they could be removed. Now, there is a company that is developing this mask that can be reused. They can be washed and reused many times. I think, I don't remember the number of times they can be used, but there is some ways to actually prevent the loss of these nanomaterials. And typically you can use the membrane, okay? And if the membrane has pores that is smaller than the nanoparticles, it will stay inside this membrane filter, okay? Okay, I have a question from Denise. How does nanomaterial recognize the virus? Which nanomaterial, Denise? Maybe Denise, you can talk if you have the microphone. Which nanomaterial you are asking me about? The one before last one. This one? Yeah. Okay. So you see, this material does not recognize the virus. You see here, what happens is a vaccine. How the vaccine work? You inject the genetic material that is very similar to the virus, but is not the virus. And you expose your body to this RNA. And when your body recognizes this RNA, it will produce the antibodies, the proteins, to recognize it when they see it inside your body and bind to them and degrade them. So this genetic material is very specific to the virus. And when this is inserted inside the cells of our body, they produce the protein, the antibodies, to recognize it. So that's how it becomes specific, okay? Okay, I have some question here again. Does that answer your question, Denise? Okay, my question from Camilo. We can use other nanoparticles like gold. Of course, Camilo, you see the idea is here to present what has been disclosed to the public. I am sure people are using gold, silver, PLGA, whatever is available. What works for one virus will work probably for this virus. But the idea here was to present the current state of what is public in terms of product, technologies, and research. But obviously, other nanomaterials could be used. Actually, Amar had few papers about gold that he shared with me, and I'm happy to share with everybody. Graphene has the same properties as any metal to kill virus. About the cost of all the nanoparticles and production, which material has less cost? Okay. Okay, very good question, Antonio. No, graphene does not have the same properties as any metal to kill virus. Graphene, as you may know a little bit, is a very thin layer of carbon material. Yeah, it can be very large in size, but is very thin. So because of that, he has some very, and also is very organized carbon structure. So because of that, he has some very specific properties. And one of the properties, because it's very thin, it can break or it can enter into the cell membrane or virus or cells. So it has a very specific, unique properties compared to other metals. How about the cost of all the nanoparticles and the production? So for example, copper oxide nanoparticles, you can make it in your kitchen. It's not so difficult and it's very cheap. Now, if you go to the graphene, of course, it's a bit more expensive. Quantum dot, gold nanoparticles. These can be a little bit more expensive. Now, if you enter into the vaccine production, it's also quite expensive. But really, at this time, again, that's what I was telling, is that the cost is a problem. But really, the most important problem is the solution that need to come out as fast as possible. And the cost is secondary at this time. And this is because of the really unique emergency situation that the world is. In other situations, for malaria, for example, well, you know, the cost is very important for malaria. But for COVID, probably the cost is not so important so far. I don't know if there are other questions.
Frank,
Yes.
could you explain again the part when you talk about the carbon quantum dots, please? I didn't understood the whole mechanism.
Okay, I can explain this a little bit more. Yeah. So in this slide, the carbon quantum dot mechanism, okay, is basically a blocking mechanism. Okay? It can be a carbon quantum dot, or it can be anything that is a little bit large and that bind to the virus surface. So what happened is that this carbon quantum dot is modified here with a functional group. This functional group bind to a receptor on the surface of this COVID virus. Yeah? And when it binds to the receptor of the COVID virus, so you basically have the virus like this, and then now you have all these quantum dots that bind on the surface. Yeah? And basically it covers the surface of the virus. And because it covers the surface of the virus, it can block the activity of this virus, because this virus now cannot enter inside the cell anymore. Yeah?
Yeah, it's similar to the present slide you showed before. This is
exactly.
The part.
It's very similar to the Phage capsid. And instead of using a protein nanoparticles, they use a quantum dot nanoparticle. That's right.
Thanks, Frank.
No problem, Camilo. Okay, how nanoparticles bind coronavirus RNA? I heard that nanoparticles are negatively charged. Maybe I did not hear. Exactly, Oscar. So that's what happens. These magnetic nanoparticles have a very high density of negatively charged, and the RNA has positively charged. So they bind to it, they capture the RNA, and with the magnet, you can remove the RNA from the solution. Yeah. Could there be any risk in inhaling the nanoparticles present in face mask? If so, will it be dangerous for health of the user? Yes, absolutely, Alejandro. That is why typically this coating of copper nanoparticles need to have two membranes on both sides to prevent to enter into the mouth and to prevent to get out of the mask. So typically there is a fiber membrane on both sides, and in the middle you have these nanoparticles. So you prevent the entry into the mouth. Okay, I guess we had many questions and I cover a lot of things. I want to thank everybody to join this class, and hopefully you learn something, and stay safe at home. And again, thank you to Amar, to Federico for all the help in this class, and to support this idea to discuss about this topic in this class. Thank you so much, everybody, and I hope I see you soon in class as well. Bye-bye, everybody.
Bye, teacher.
Bye, Frank.
Bye, teacher. Thank you.